Welcome, I am Gabriela Merino and I will present you integrating multiple information sources for protein function prediction with end-to-end deep learning. The most precise way for assigning functions to proteins is through manual curation based on experimental evidence. However, it is extremely expensive and time-consuming. So, for this reason, the community have been extensively working on the development of computational models able to process data from multiple sources to automatically predict protein functions. Especially for those proteins that have not been previously annotated, known as no knowledge or NK proteins, integrating knowledge from multiple sources is a key to develop precise models. Although many computational models for predicting gene ontology terms describing protein functions have been developed, their performance is still subject to improvement, even more for those NK proteins. Here we propose DeeProtGO, a deep learning model for the automatic function prediction of gene ontology terms by integrating protein knowledge. It consists of two blocks involving several input encoding subnetworks and a classification subnetwork. The input subnetworks are aimed to process each input data in a separate way and encode it into a learned feature space. These learned features are then integrated into a single vector that is used as input of the classification subnetwork. This subnetwork has 6 fully connected layers aimed to predict the set of Go terms for each protein. The last 4 layers model the output taking into account the hierarchy of gene ontology from the most general terms to the most specific ones. The model is trained in an end-to-end fashion using the sum of the binary cross-entropy laws of these output layers as a cost function and the Adam optimizer. DeeProtGO was trained and tested considering the 3 time points of kappa3. T-1 when training and target proteins were released, T0 the deadline for submitting targets predictions, and T1 when benchmark proteins were collected. The dataset for evaluating DeeProtGO was obtained by selecting those no-knowledge proteins in the kappa3 benchmark dataset. To train DeeProtGO, we considered from T-1 to T0 as the annotation period for training proteins. Since kappa3 training dataset do not include no-knowledge proteins, we obtained them from the corresponding versions of UniProtKB SwissProt. However, these proteins were very few for training precise models. Thus, we designed a data augmentation strategy. For each sub-ontology of gene ontology, we analyzed the set of kappa3 training proteins and selected those that, for this sub-ontology, are no knowledge. It means that this protein did not have annotations at T-1 for this sub-ontology, but have gained at least one annotation during the growth period that we defined for training data. When we are trying to predict functions of no-knowledge proteins, the unique information that we have for most of them is their sequence and the organisms from which they belong. Thus, the input data that we used for DeeProtGO was based on this information. So, input features from sequences were obtained by embeddings of length 1024 generated with StackVec and by defining a protein similarity measure based on the pairwise sequence edit distance. Organism taxons were also used as input by representing that with a one-hot encoding vector. So, considering the 3 subontology of Go and the 2 taxonomic kingdoms, Prokarya and Eukarya, we trained 6 different DeeProtGO models, each of them involving at the most 3 input encoding subnetworks for learning the protein features that were then used for predicting their functional annotations. All these models were trained under a cross-validation scheme involving 3 folds and a partition of the training data into 70, 10, and 20% for training, validation, and testing respectively. Hyperparameters were fine-tuned by grid search, focusing the optimization process into the dropout probability, the batch size, the number of neurons in the hidden layers, and in the learning rate. The number of epochs was optimized using an early stopping strategy, and all model performances were compared and assessed using the Fmax precision and recall defined and used for the kappa challenges. The results obtained during DeeProtGO training for the best configuration of the 6 models are shown in this bar plot. Thus, for each subontology and each taxonomic kingdom, the filled bar represents the reached Fmax and its color the integrated inputs. In addition, unfilled black and blue bars indicate the corresponding recall and precision. So, as you can notice here, for biological processes, the Fmax reached were 0.49 and 0.39 for Prokarya and Eukarya, respectively. The Fmax for predicting cellular components was 0.70 in the 2 considered models, Prokarya and Eukarya. Meanwhile, in the case of predicting molecular functions, Go terms, the Fmax reached were 0.64 for prokaryote models and 0.82 for eukaryotic models. In most cases, the precision reached by DeeProtGO was higher than the corresponding recall. Our results also indicated that integrated sequencing and taxon data was the best for doing predictions in biological processes and cellular components. Meanwhile, in the case of predicting molecular function terms, using only the embedding of the sequences was enough for achieving good results. The performance measures DeeProtGO reached in the benchmark datasets are presented in this table. In summary, the overall Fmax that DeeProtGO reached predicting biological processes terms was 0.34. In the case of cellular component predictions and molecular function predictions, the overall Fmax were 0.55. Same as during training, these models showed, in most cases, a higher precision than the corresponding recall. The overall Fmax scores achieved by DeeProtGO were compared against those reported for the top-performing methods on the CAFA3 challenge. As it can be observed here, DeeProtGO has performed in the top 5 CAFA3 methods for both biological processes and cellular components. Even more, it is the second best method when predicting molecular function terms of no-knowledge proteins with high precision, enhancing the discovery of new functions. So concluding, DeeProtGO performance is competitive to the top 5 methods of CAFA3. Even more, DeeProtGO is the second best method in molecular function. Our results reveal that as more input data were integrated, better scores were reached for both biological processes and cellular components, and that also data augmentation allowed to train precise deep learning models. Thus, in summary, deep learning models integrating multi-source data are a promising tool for protein function prediction. Thank you to all the team that participated in this work, and thank you for your attention.